Hello, thank you for joining me today for my first ever cellular molecular biology e-report. Today we are going to go over the concepts of molecular mechanisms of the cell and the central dogma of biology. So before we discuss chromatin compaction, I think it is very important to note that the structure and function of DNA allows for it to be the central molecule of life. Because of its chemical components, DNA can actually hold on to all the information ever needed to create life, and then it can replicate that information to sustain it. This is core to understanding the central dogma of biology, which is the process of DNA transcription to mRNA and mRNA translation to proteins. So we have approximately 6 feet of DNA in every cell of our bodies, which makes up the human genome in every cell. All of this information must be packaged or compacted properly in order to ensure that the information can be condensed into the small space that is the nucleus, but also so that all of the information required for DNA replication or mitosis is accessible to the cell in an organized way. In bacteria cells, this process is actually accomplished more simply with supercoiling because the bacterial genome is actually circular. In eukaryotes, however, chromosomes is the term used to describe the long packaged linear DNA molecules that are compacted by looping, coiling, and spooling in an organized way. Pictured here is actually a beautiful image of a mitotic chromosome that has been compacted and condensed to its mitotic form for cell division. We have 46 chromosomes, 23 pairs, and each one can simply be stated to be a single very long molecule wrapped up and compacted by proteins. DNA is its most compacted or dense during mitosis at nearly 10,000 times compaction, and this is important because chromosomes are dealt out in a highly specific way during cell division. It is during interphase that these chromosomes are most relaxed. Chromatin compaction is a highly conserved process in all eukaryotic cells. This begins with the naked, uniform, protein-free DNA that we are now so familiar with. The first level of compaction is characterized by nucleosomes, which are pictured here. A nucleosome is comprised of a series of 4 histone proteins, H2A, H2B, H3, and H4. These 4 proteins fit together to form a protein spool. A histone octamer structure, which is pictured here in entirety, is formed when 2 of these protein spools, so double each, H2A, H2A, H2B, H2B, H3, H3, H4, H4, are present with the naked DNA wrapped around the spools that are joined together like thread. This compaction around the octamer structure occurs because of the relatively negative charge of DNA compared to the positively charged histone octamer to form a nucleosome. DNA is relatively negative because of its phosphate groups. The protein H1, which is pictured here separately from the nucleosome, is on the outside of the nucleosome. Although it is a histoprotein, it's not a part of the octamer structure because it has a specialized function. The H1 protein is responsible for kinking the linker DNA, right here, pictured in purple, that is in between each nucleosome in order to ensure that the next level of compaction occurs, which is the 30 nanometer fiber. So the H1 histone protein manipulates the linker DNA by kinking it into coils, much like wrapping a beaded pearl necklace around your finger over and over again. This coiling results in the uniform structure pictured here and here, where the H1 histone proteins are central to the coiling process by means of the linker DNA in between the nucleosomes. So once this level of compaction into the 30 nanometer fiber is completed, it is largely unknown actually how DNA chromosome compaction is accomplished in order to produce the final end product, a mitotic chromosome. It is believed that the proteins wrap around the DNA and scaffold on top and around one another in an organized way geared by molecular forces that are unknown in order to produce the folded and dense structure of a mitotic chromosome. As you can see from this diagram here, we see the nucleosome structure produced by histone proteins, the H1 manipulating linker DNA to create the 30 nanometer fiber, and then the protein scaffolding discussed, until we reach the final end product, which is a mitotic chromosome. Localized unwrapping of DNA is actually essential or necessary to utilize DNA sequences, or genes, when it is needed by the cell while still maintaining the dense level of compaction or packaging in the rest of the chromosome. A cell can utilize several strategies in order to gain access to tightly packaged but needed sequences of DNA. These methods include chromatin remodeling complexes, which through hydrolysis simply use brute force to push the needed DNA out of a chromosome, which temporarily physically modifies the structure of the compacted DNA section. What is most important to discuss, though, is the role of acetylation and deacetylation in the locking or unlocking of needed sequences within the nucleosomes. There are histone tails that project out of each nucleosome that are rich in the amino acid lysine, which carries a native positive charge on its side chain that can be interacted with or manipulated through acetylation or deacetylation. These here pictured are the histone tails that are rich in lysine. When lysine histone tails are acetylated, their positive charge is neutralized by histone acetyltransferases, or HATs here. Acetylation by HATs actually facilitates the loosening of a nucleosome in order to provide access to the DNA sequences. When lysine histone tails are deacetylated, their positive charge is restored by histone deacetylation complexes, or HDACs, that lock nucleosomes in again to inhibit access to DNA. These mechanisms are needed in order to facilitate responses to change in a cell when it is needed for regulation at the level of chromatin. Because we know DNA to be this beautiful, uniform molecular structure that is responsible for carrying the information for all life, it makes sense that the mechanism for replicating that DNA when it is needed would be simply beautiful as well. The process of replication ensures that hurtful mutations do not interfere with the dutiful copying of DNA. It is a fast and almost perfect process. The patterns and rules of base pairing between nucleotide bases allows for this almost perfect process of replication because each strand of singular DNA acts as a template for a complementary strand. This is known as semi-conservative replication. The origins of replication, where it starts, are bound by initiator proteins, where there are only a few of many hydrogen bonds are broken. Only a few hydrogen bond breaks are needed in order for replication to occur at something called a replication bubble, or the replication fork. At the replication fork, the double strands are pulled apart, as we see here. What is cool about the replication fork is that it occurs bidirectionally, which means that DNA synthesis occurs at both sides of the replication bubble by forming two complementary strands for each of the two separated strands. So I chose to include this diagram because I really enjoy the simplicity of the way that this is set up to demonstrate the process of DNA replication at the replication fork. Here we see an intact DNA double helix that's separated by initiator proteins to break the hydrogen bonds between them at the replication fork. From there, each strand of DNA acts as a template for another to create a new complementary strand running anti-parallel. This replication produces two identical DNA double helices, each with one new and one old strand. The protein DNA polymerase is responsible not only for creating phosphodiester bonds between 5' phosphate groups and 3' hydroxyl groups, but also for the synthesis and processing of deoxynucleotide triphosphates, or DNTPs, which carry the energy needed to steer on DNA replication through the cleavage of two phosphates off of the DNTPs to create a nucleotide base, which makes up the complementary strands. This DNA polymerase is held in place on the DNA strand by a clamp protein, which is placed there by a clamp loader protein that ensures that DNA polymerase does not leave the site of replication by floating away. The replication bubble, or its fork, maintains its integrity by DNA helicase, which pulls apart the strands in front of DNA polymerase continually through the ATP, while single-stranded binding proteins keep the single strand from reassociating with its original complementary strand during replication. Replication by DNA polymerase can occur at the rate of 100 bases a second when it's held down by a clamping protein, which is just amazing. While DNA polymerase is synthesizing DNA, it can also proofread and correct incorrect sequences that it creates on the complementary strand itself, and it does this by touch. due to the uniformity required in DNA structure. At the replication fork, one strand is synthesized with the leading strand 5' to 3' continuously, as we see here. Nice and smooth. But because of the rules of base pairing and antiparallelism in the DNA structure, there is a lagging strand that must replicate discontinuously at the replication fork, as pictured here. Fragments. This means it must be done in the same way, 5' to 3', but in fragments. These fragments are now called Okazaki fragments, which are synthesized asymmetrically and then later joined to create a full complementary strand, which we will go over. So DNA polymerase actually can't make DNA on its own without the aid of RNA primers, which are pictured here in red. RNA has the capability to make complementary strands de novo, which means right on the spot. Each RNA primer contains 10 bases that are added by DNA primase. The leading strand needs only one RNA primer to begin the replication of its sequence continuously, whereas the lagging strand needs many RNA primers in order to create many fragments that are later pulled together into a full strand. There are 3 enzymes that are essential for joining the Okazaki fragments together in the end on the lagging strand. This includes the 3' end enzyme that chews up the RNA primer placed there, the repair polymerase that replaces the RNA primer with DNA instead, and then DNA ligase, pictured here, which joins the fragments together through phosphodiester bonds. All of these proteins we've described previously during the opening of the replication fork, the synthesis of DNA itself, and the joining of discontinuous fragments on the lagging strand, all form a giant replication machine that works together to replicate DNA at the replication fork bidirectionally and simultaneously at varying points in DNA. So the repair mechanisms for DNA repair of mutations go to great lengths to ensure that all of our mutations are minimized for us. These evolved repair mechanisms are pivotal to our cell's survival, as a mutation at the level of DNA would mutate all of the information that would be utilized during transcription or translation. So DNA mismatch repair is a mutation repair mechanism that is dedicated to fixing the mistakes made by DNA polymerase that were not caught by its proofreading function that we discussed earlier during replication. DNA mismatch repair simply repairs bases that are not matched to their correct nucleotide. One might ask, how does DNA mismatch repair even know which strand is the one that needs to be corrected? DNA polymerase can proofread the strand it just made, but DNA mismatch repair can actually determine which DNA strand is actually old or new, incorrect or correct, through hemimethylated strands, where the older original strand will be methylated as a biomarker or a flag that it is indeed the older strand. The unmethylated mutation sequence is cut out and the incorrect base is replaced with new ones, and DNA ligase seals the gap created in the DNA backbone. The unmethylated strand is repaired and then becomes methylated as well to be marked as an old strand. Double-strand breaks occur as a result of DNA severing into 2 pieces, which is actually quite dramatic and scary, and it can be life-ending for a cell. This can occur due to radiation or free radicals, which is why it can be so devastating to a cell if it does not utilize a repair mechanism to fix it somehow. This repair can occur through 2 mechanisms: non-homologous recombination or homologous recombination. Non-homologous recombination results in the blunting of severed strands and simply gluing or ligasing them back together haphazardly and deleting the sequences that were lost as a result. And this could actually result in a loss of function mutation. So here we picture a non-homologous end joining where there was high energy radiation that severed the 2 strands, and they're just haphazardly glued back together in order to preserve that structure. Homologous recombination, however, is a little more complicated, but that also means it's more effective at repairing double-strand breaks if it is possible for the cell to accomplish. Homologous recombination requires that a homologous DNA molecule is nearby to the double-strand break. The homologous DNA molecule acts as a guide for the repair, which results in the strand invasion. Strand invasion, as pictured here, involves nuclease, which chews back one strand, creates a branch point, as pictured here, that creates a fork by DNA helicase in the homologous intact unbroken DNA molecule so that This branch point can access the homologous chromosome. The strands cross together and elongate and are nicked into two intact molecules afterwards, which essentially recombined and borrowed information from one another in order to preserve similar nucleotide sequences on the damaged DNA molecule. If this is possible for the cell to accomplish, it will, but there is no guarantee that the homologous chromosome needed for this repair will be nearby for the cell to access. So the next topic we're going to discuss is the assembly and purpose of the pre-initiation complex, or the PIC, in transcription. It would be easy to think that RNA polymerase, all on its own, transcribes DNA into mRNA simply. It already does that one big thing, so it might as well do it on its own, right? Wrong. So in eukaryotic cells, this is a complicated way to initiate transcription through the PIC, but in bacteria cells, the process of recruiting and positioning RNA polymerase on the promoter is accomplished simply by a sigma factor that binds and disassociates as it is needed. This is because transcription and translation occur simultaneously in prokaryotic cells because they just are in the cytoplasm. There is necessarily no nucleus that separates transcription from the process of translation. But in eukaryotes, the process of recruiting an RNA polymerase is initiated by, oddly enough, a pre-initiation complex, or PIC. Our RNA polymerases cannot initiate transcription unaided or alone. The pre-initiation complex is a group of general transcription factors, or GTFs, that allow for RNA polymerase to be recruited to the promoter region of DNA in order to position it and send it down the length of a gene during transcription from DNA to mRNA sequences. There are several GTFs that are named based on a Roman numeral and a single letter in order to discern between them. So pictured here, the first GTF to bind and recognize the promoter is TF2D, right here, which includes a protein subunit called TPB, which is not pictured here, but that TPB it stands for Tata binding protein that, what do you know, binds to the promoter Tata box sequence that is rich in T's and A's, or thymines and adenines, as we call now the Tata box. It kinks the Tata box, which makes it unmistakably recognized by the following GTFs that are recruited to the pre-initiation complex after TF2D binds. One each GTF is recruited to the pre-initiation complex. Transcription of the RNA polymerase can begin through the positioning, separation of a double-stranded DNA, and launching of the RNA polymerase down the gene. The PIC dissociates when transcription has ended. So here is another diagram that just shows the process of recruitment of the pre-initiation complex. Here is the promoter on the gene, the Tata box, rich in T's and A's, that TF2D recognizes. The Tata binding protein is within, binds to the Tata box, and each of the other GTFs are recruited, such as TF2A, TF2B, and then the following TF2E, TF2H, TF2F, until RNA polymerase can be recruited onto it. After mRNA has been transcribed by RNA polymerase, it must be processed in a way that marks it as a complete and ready molecule to enter the cytoplasm through the nuclear pore for translation to subsequently occur. The mRNA will only be ready if it has been capped, tailed, spliced, and circularized. Oh my! Eukaryotic mRNA processing is horribly complex for the reason that transcription and translation are processes separated by the nuclear membrane and cytoplasm. This ensures some quality control, though, in our eukaryotic cells for our molecular mechanisms. So first, the mRNA receives a cap that is comprised of a methylated guanine attached in a weird way that is added to the 5' end of the mRNA in order to mark the mRNA cap as the beginning of that sequence. Here's a simplistic drawing of a mature mRNA with a methylated guanine attached as the 5' cap. After this, mRNA receives a poly-A tail, which is usually hundreds of adenines long, as seen here. This is added to the 3' end of the mRNA to mark it as the end. Both of these modifications made to the mRNA mark it as a stable molecule, which allows it to be ready to leave the nucleus. Proteins are then recruited to the cap binding protein, and the poly A binding protein, presumably, that interact with one another to circularize the mRNA. But before this happens, however, mRNA must be spliced, which is pictured below here. Splicing ensures that needed sequences, or exons, are separated and then spliced away from introns, which are pictured here. Introns are cut from the mRNA sequence. Although both exons and introns are transcribed into mRNA from DNA, only the exons are needed for translation of proteins and are spliced together after everything's been capped, tailed, and circularized. Once these processes are all complete, the mRNA is ready to leave the nucleus. Alternative splicing allows for the modulation of mRNA processing, meaning that there is no rule stating that every exon in an mRNA sequence must be kept for translation, but instead any varying number of those exons may be spliced in or out to create a variety of translated proteins from that sequence. So here is another diagram that represents mRNA processing, starting with DNA being transcribed into an mRNA with an mRNA cap, the methylated guanine, a poly A tail of hundreds of adenines, that is then spliced into exons and introns, where the introns are then left out to be lariats, and the exons are actually spliced and glued back together in order to be ready to exit the nuclear membrane through a nuclear pore. And this mRNA will be circularized, marking that it's ready for transport out of the nucleus into the cytoplasm for translation. So transcription, which transformed DNA into mRNA, an easy, portable, and usable molecule pictured right here, that can be translated into the language of proteins, which is amino acids. Below that is the simplest concept of translation represented by a ribosome using mRNA and tRNA molecules to create chains of amino acids in order to form more complex proteins. To be honest, the longer I looked at and thought about ribosomes, the more I realized that ribosomes are always represented in a way that makes them look like Super Mario mushrooms that you can use to give you a boost in Mario Kart. Besides the point, though, that proteins are really central to our lives and our sustained functions. Translation, as we have named it, is merely a representation of a how-to on building proteins in order for us to live. So it was heavily debated for a long time as to how 4 nucleotide bases, adenine, cytosine, guanine, and uracil, could somehow come together and translate into 20 functional amino acids definitively. Scientists discovered that a series of 3 nucleotides, or a codon, is required to code for the translation and production of 1 amino acid. This is now represented by the genetic code, which serves as a full translation guide for understanding which codons translate to which amino acids for all living things. The open reading frame is the first codon sequence to be read in a series of sequences in an mRNA molecule, and the open reading frame is the first and is always AUG, or the start codon. The sequence of mRNA codons always ends with a stop codon, which signals the completion of the protein being translated. mRNAs have no way to recruit amino acids for translation directly on its own. It requires another RNA molecule called tRNA that bridges the gap between mRNA and amino acids, as pictured here. mRNA, amino acid, and in between, the tRNA. The tRNA contains an anticodon, which is a complementary sequence of 3 nucleotides that codes for that specific amino acid in relationship to the mRNA codon. The sequences are always read 5' to 3'. As pictured here, although the codon is AUG and the anticodon appears to be UAC intuitively, it is actually read CAU because this is 5' to 3' anti-parallel. tRNAs carry their corresponding amino acid on its end, and the tRNA intramolecularly base pairs with itself in order to create a cloverleaf type shape, or a T. that further folds into an L shape, where the anticodon end and amino acid end are the only ends exposed. So if you see here, this is the most basic structure of a tRNA, where it is intermolecularly base pairing with itself because it can. It's an RNA. And here is the anticodon sequence that binds to directly the mRNA codon sequence. Up here, the amino acid binds through a process called charging, which we will discuss. But here, this cloverleaf structure folds into an L shape where only the amino acid at the top is exposed and the anticodon at the bottom is exposed. tRNA charging occurs in order to attach the proper amino acid to the end of the tRNA with its corresponding anticodon, and this is accomplished by aminoacyl tRNA synthetase, which recognizes the sequence in the anticodon to determine the attachment point. So, although tRNA facilitates the direct translation of mRNA sequences into amino acids, it is the true translator. Ribosomes are huge protein RNA complexes where protein synthesis occurs within. Ribosomes are made up of approximately 82 proteins and 4 long RNA molecules that act as a ribozyme, which are characterized by a large subunit that builds peptide bonds between the amino acids and a small subunit that matches the tRNA to its corresponding mRNA codon to anticodon. Here pictured is the small ribosomal unit, which pairs the mRNA to the anticodon on the tRNA, and a large ribosomal unit, which houses the voids in which amino acids are joined together by peptide bonds. These two subunits are sandwiched together around the mRNA, and they scan for AUG, the start codon, in order to initiate the process of translation. In the large subunit of the ribosome, there are 3 voids known as the A, P, and E sites that can perfectly accommodate each tRNA that is interacting with the mRNA sandwich within. The A site accommodates the aminoacyl tRNAs, or the tRNAs that are dropping off their amino acids with them still attached and charged. The P site accommodates the peptidyl tRNAs, the tRNAs that are creating peptide bonds with their amino acid to the amino acid that enters the A site. And then the E site is the site at which the tRNA finished with translation exits. It is important to note that at any one time there can only be 2 tRNAs within a ribosome. In eukaryotic cells, every translation starts with a tRNA that enters the ribosome through to the P site instead of the A site at first, because it is the first in a sequence of many tRNAs that must create peptide bonds with the following paired amino acids that are on the incoming tRNAs. That initiator tRNA, which is pictured here with the sequence CAU on the anticodon that pairs to UAG, holds the charged amino acid methionine, which starts the sequence. This initiator tRNA associates with a free small ribosomal unit, pictured here, through EIFs, or eukaryotic transcription factors, at the 5' cap of the mRNA, and recruits all of the necessary players of translation, while the initiator tRNA searches for AUG that corresponds to it along the mRNA. Once it is bound and associated with the AUG codon, the initiator tRNA is placed in the P site of the large ribosomal unit as it joins, and it is recruited to the mRNA as well to form this initiation complex to begin translation. Another tRNA will enter the A site of the ribosome and bind with the codon of the mRNA through base pairing that is next following AUG. The start codon AUG that codes with the anticodon CAU, which carries in the methionine, then creates a peptide bond with the next amino acid on the following tRNA in the A site. Then the amino acid is released from the tRNA complex now that it is chained together. As another tRNA enters, and so on and so forth, One tRNA always leaves through the E site, as there can only ever be 2 RNAs in a ribosome at any given time. This process will continue until the mRNA reaches the stop codon, no matter how many proteins need to be made within that amino acid chain. There are no tRNAs that actually bind to stop codons with an amino acid, so a release factor enters the ribosome, and that cleaves the bond of tRNAs from the amino acids and releases the peptide protein from the ribosome entirely. From here, the protein has been synthesized and prepared to function the way it was meant to elsewhere. So wobble base pairing lets the same tRNA recognize multiple codons for the amino acid that it carries. And this is an important function in translation because the rules of wobble pairing ensure that a tRNA does not bind to the wrong codon if there's an atypical base pair. For example, tRNA carrying phenylalanine can actually base pair with 2 different sequences in case something were to go wrong. It carries the sequence GAA on the anticodon, but can either base pair with a UUC or a UUU in case anything were to go wrong, so that the same amino acid ends up in the chain that it was intended to for the protein that's being created during translation. And although these tRNAs are distorted a bit by having to conform to another codon through wobbling, it is allowed to, in order to ensure that the mistakes made by mRNAs will actually result in the same protein production. So it's a pretty cool function. Although DNA contains all of the information needed to create and sustain life, like I've mentioned before, a cell cannot simply function based on the presence of that information, or the absence for that matter. The complexities of eukaryotic gene expression allow for the careful, precise control of the life of a cell through the regulation of proteins, which govern all of our function. In order to exist, cells must constantly respond to change that is occurring in your environment, during your development, and especially within the body at all times. So, although regulation and eukaryotic gene expression can be controlled at any point within the central dogma of biology, meaning at the level of transcription, at the level of processing, through translation, through the regulation of the actual active protein, it is actually most commonly regulated prior to the initiation of transcription, right about here. And this is because it is the least energetically expensive for the cell, or it doesn't require a lot of work in order to do, because if a protein has already been created as a result of transcription, mRNA processing, translation, and the processing of that protein, your cell has already gone through all the effort to create this protein, and it must take a lot of energy in order to ensure that that protein doesn't function at a time when it is not needed, or that it would hinder the cell's function. This is my favorite diagram included in this presentation. Eukaryotes require a high level of refined, precise gene regulation in order to maintain synchronized, coordinated, interdependent systems of life. Sounds crazy, right? Genetic transcription factors, or GTFs, which should sound familiar, can bind to upstream or downstream enhancers that are great distances away from the actual gene that they regulate. As you can see here, there are many spaces in between the DNA and the regulators that are associated with this gene over here. And they regulate many of our genes that are not regulated really only by one factor. It would be more simple if one factor controlled one gene, but more often than not, there are many activators and repressors that control the action or transcription of that one gene. This is called combinatorial control, which allows for several regulatory proteins to precisely control the expression of a gene based on a response from multiple signals within the cell, kind of like a democracy. There are many votes that come in from all around the DNA that contribute to the final decision to either express or not express that particular gene. And in eukaryotes, gene expression is a committee decision, very much so, based on the votes from dozens of activators and repressors, which are just shown here in brief. So the individual responses and signals coming from these enhancers physically interact with something called the mediator complex, which was shown previously here. Attached to the Tata box, RNA polymerase associated with the promoter at the start of transcription. The mediator complex can summate one single signal from several of these enhancers, activators, repressors that can be relayed to the promoter to either repress or activate transcription at this level. The mediator complex is a conglomeration of many proteins that create a complex that interacts with all those enhancers at all the genes that are non-housing. And when we say housing genes are upkept in all cells. They're required for all cell function, but there are many genes that are required in many different types of cells, and this is very important in cell differentiation. But the mediator does just that. It mediates combinatorial control. Although the subunits located on each mediator complex varies between and within species, the core concept has been evolutionarily conserved among all eukaryotes as a definitive function of combinatory control. So cool. So from this picture here, you can see that there are many, many proteins that contribute to this massive conglomeration of proteins that is the mediator complex. And this should be familiar from earlier in the eReport where we discussed transcription and the PIC or pre-initiation complex recruiting RNA polymerase for the act of transcription, here with all of our transcription factors on the Tata box. The function of RNA interference, or RNAi, is to permanently silence certain sequences of mRNA prior to translation as a form of gene regulation. It is theorized that it evolved as a mechanism to handle primitive viral infections within the cell. Gene silencing is important in order to ensure that certain genes are not expressed in cells that cannot benefit from that particular gene's activation at that time or ever after. Gene silencing becomes particularly important during cell differentiation. For example, you do not want your liver cells to be expressing genes that are found in neural cells. Gene silencing can occur through many mechanisms such as methylation or downregulation prior to even transcription like we've discussed before, but one mechanism that we will be discussing in detail is RNA interference. RNAi ensures that certain double-stranded RNAs are not translated into proteins. We often associate double-stranded RNAs with viruses. Double-stranded RNA, or commonly known as an invading virus in eukaryotes, can be bound or chopped up by an enzyme called the dicer enzyme, which are proteins that can chop up double-stranded RNA into pieces, which results in many, many siRNAs, or small interfering RNAs, to be produced. These siRNAs can base pair with any complementary RNA sequence or strand in the cell through RNA-induced silencing complex, or RISC, which is pictured here, which is really cool and scary because it has the ability to destroy those complementary strands that the siRNAs base pair with on demand, as long as the complementary siRNAs are actually bound to it. So this mechanism, through the function of RISC and siRNAs, can be particularly useful when you want a sequence of coding RNA to be destroyed permanently, as it is bound to the RISC and searches endlessly for those sequences to destroy. And in mammals, particularly, not prokaryotes, not all eukaryotes, but in mammals, this process can even be amplification, through something called RDRP, or RNA-dependent RNA polymerase. RDRP functions by taking the siRNAs that bind to RISC, reading it, and replicating it over and over, as seen here, to create more and more RISC complexes so that any mRNA molecule carrying that particular sequence can be destroyed and more quickly. So as you can see, the dicer dices up the siRNAs, but what happens instead is that RDRP steals away those sequences of siRNAs from the RISC complexes and develops them over and over again into secondary siRNAs that create more and more in order to amplify the process of destroying those needed genes that need to be gene silenced. RNAi has actually been exploited for the use of laboratory research in order to prevent protein expression of targeted genes in a controlled setting. So by expressing created siRNA molecules or synthesized siRNA molecules that have actually been made in the laboratory, there can be almost an immediate stop to protein synthesis in the laboratory as a result. So if they're working with a particular protein that they need to silence in order to get a result that they're looking for, then they can use these synthesized silencer siRNAs or stealth siRNAs. In lab applications, artificial genes can also be made so that when they are transcribed, they form stem loops, which stem is a double-stranded RNA. They intermolecularly based pair, and these stem loops form through that base pairing between the RNA bases. They look much like siRNAs, and these artificially created double-stranded RNAs can be transcribed and used through the same process of RNA interference. So this final topic concludes the eReport for this first part of cellular and molecular biology, in which we discuss the entirety of the central dogma of biology and the many intricacies that are involved with that. I hope you enjoyed watching. Thank you.